Good morning, one and all. In this session, we'll be learning protein identification and interaction and much more details connected to the mass spectroscopy. But before we go for it, let's understand this entire strategy in a nutshell so that we can go deep inside it. Uh, Whenever we are working on a protein, any protein, we normally get several different proteins in the lab when we are working on, and we normally want to identify the function or what are different functions that are encoded in this particular protein. So to do it, what we normally do, we check out the take the sequence, put on blast, search on different servers. We normally do it, but before it all turned up, the mass spectroscopy's technology turned up, which led into a revolutionary set of ideas coming up in the picture in research field. What those people did, their main concern was they using an electric field, they were able to generate fragments on the protein sequence and then getting the segment size. So whenever you are working on protein, you take the cellular sample, extract the protein, you break down the protein into segments using proteases, as description shown here. Then you do the mass spectroscopy. We'll be knowing the details shortly. Then you search it in the server, analyze the sequences. Okay. Then once you work out, see, we also build the phylogenetic profiling and protein-protein interaction networks through which you learned how many different proteins are evolving together, which is an interesting concept I'm linking to this set of slides. So to understand is how many different proteins in a cell function together. So if a mutation turns up in one protein, a corresponding complementary mutation turns up in the other protein as well, so that the corresponding function go as such without any troubles. Okay, that is termed as co-evolving protein pairs, or co-evolution is going on, which is an important concept in applied bioinformatics. Okay. So when I get this protein sequence, the entire question is how to how to understand the protein function or how to identify what is this protein all about. Like if I have a protein, normally I may get its protein sequence, like molecular weight, physical or chemical characteristics, like uh, but nothing more than that. So the entire question or the screen is how to search the function or how to hunt various different peptides which are present there, which may be crucial or which may be only present in this protein as a representative. Okay, so as a fingerprint. So identifying means presence, quantity, identifying the proteins exactly from the sample. What is this all about? So mass spectroscopy is technique is a powerful strategy, a powerful technology that measure the masses and relative abundance of the molecules or the fragments. So the basic steps of mass spectroscopy strategies technologies, you need to take the cell lysate, run the complete cell lysate using a 2D page. Then as our objective is to screen or identify different fragments, crucial fragments protein present in a protein, encoded in a protein. So what is our entire part? We need to chop it using protease, of course. So we normally use any trypsin or anything like that. We normally prefer trypsin. We'll discuss the reasons. Okay. Then as the protein fragments are coming, we need to see that no other contamination turns up, because it will spoil the entire result. So then we can run the fragments using the multi-top is an instrument, matrix assisted laser detection ionization time of flight. Okay, and the first one is the machine, and TF is the time of flight is the strategy to capture the like uh, entire set of fragments used under a magnetic field on a detector. So see, like this is what exactly I was saying as this mass spectroscopy is using like done through this multi-tube machine. Like here under the electric field, like the sample matrix when you're putting the sample here, all the like broken up peptide fragments run under the laser C here using the charge. Like heavier atoms like go slower than the lighter ones. Okay, because they cannot accelerate drastically. Okay, so see, once it reaches here, the detector is there. It all works out using a time of flight which captures the time, the motion which is there, and accordingly, like based on the like a mass upon the charge, Q by M ratio of different like amino acids, we can easily check out what is the overall fragment which is coming up. Okay, see, it works on the quantification only of the base of like charge which is like imparted under like a particular magnetic field for the given peptide segment. So this got the Nobel Prize see, in 2002. The Japanese Koichi Tanaka along with John Fenn. See, the development of soft desorption ionization methods for mass spectroscopy of biological macromolecules. Advantage is here many number of protein fragments. Even if two or three proteins are present together, we can identify that this mixture contains two or three proteins because it will have like a crucial signature fingerprints which are conserved only in these two or three different protein sequences. We can exactly find out. See? Trypsin cuts C terminal of lysine or arginine. So now you know like why we are using trypsin. Because it cuts at a particular place, particular position. So if we know that, we can exactly generate the similar fragments in the system as well, so as to compare the masses. So now you understand like when we are doing the things in the lab, we can exactly like validate the results in the system as well. Okay, so that is the experiment going rightly or not. Okay, what kind of a result am I expecting for this sample? Because its sequence goes like this. Okay, that's the entire advantage here. Okay, you see, for these seconds I'm getting a result like this. This is more detail you can see here. If this is the part, I can zoom it here further. See, see this one in more detail. See, this is termed as peptide mass fingerprinting, PMF. Was for a protein, like specific protein fragments are coming up, and for each of these protein fragment, you will have the Q by M, like charge upon mass ratio, and for there's some specific counts as well. How many such fragments are present in the given sample? Okay, because they'll all move together under the electric field. So charge upon mass, M by Z, see, mass upon charge ratio, see, the mass upon charge ratio, see, it works here. Understand students, one more thing like a charge by mass ratio normally used in like agarose electrophoresis or like electrophoretic instruments where like under an electric field like when you apply some solvent or something like molecules move charge upon mass q by m. Here it's m by z mass upon that charge. Okay, so that is the specific concept which is coming up. So there also we use the electrophoretic mobility depending on the charge, okay, or no mass. Here also the similar concept is stand up, but we reverse the part. Okay, see, peptide mass fingerprinting. See, the PMF which I was saying. See, trypsin is a highly specific. Cuts after K and R means lysine and arginine, except if followed by P protein as such. Okay. We do the same thing in system in silico digest and actually do it in the lab, so we can like compare the results. If both the results exactly go like overlapping each other, then the things are correct. See, So if you do the database search, de novo versus database search, with computer like in the lab you're doing, and when you're checking it in the system, see database of known peptides, this is experimental data, you get up the fragment like this. Now a small question, what is the probability that this answer is search is correct? I'll give you the answer, see, you can think. So the answer lies in these steps. See, if you do a protein separation, digestion using trypsin, mass spectroscopy, in silico digest, then you compare the results. Okay, then you compare the results. So here, like, but to search it in the database, we require several different like databases like Expesi, Profound, Mascot. These are specifically developed databases only for this purpose. See, like, besides this basic search, I'm using a probability score also. Termed as a like p-value which is there. Normal. It's not a statistical p-value. It's a simple probability score. See, what is the chance of that exactly the same part is there, or if this is there, see, this belongs to this part. So many times what happens rather than a complete conserved fragment present in a like a given peptide fragment, only a small fragment is there. Rather than this complete part being there, only this is there. But you realize that this exactly overlaps. So this should be correct. Yes. So this is a probability score. But now if you consider that statistically, tell me what is the probability that this threonine is present at first position? 1 by 20 because 20 is normal amino acids. I'm taking only natural amino acids. So considering only the natural amino acids, the chances are 1 by 20. When T and P I'm taking together, what is the probability? 1 by 20 raised to 2. So likewise, suppose if 10 is there, 1 by 20 raised to 10. So now like one in such a like a large amount of large amount of sequences, only one chance will be there that you will get the fragment like this. So see, such a high probability is there like even when the probability is gone 0.65, 0.76, I still consider it to be like statistically correct. Are you understanding? See, just correlate the part. Like 1 by 20 out of 20 amino acids, if one is there, and you multiply it over like put the like multiply that number with the keep on multiplying for the length number of amino acids which is there in the like protein or the length of the protein fragment. Okay, so if you consider that part, okay, see, I'll get that part. This is a very crucial part. Okay, now see, to continue it further, if I just talk about the discrimination power of this mass spectroscopy for two very closely like, related protein sequences. See here I'm doing alcohol dehydrogenase 7, 374 amino acid belonging to human only. Okay. It is giving me 26 peptides greater than 500 Dalton. I'm not taking smaller peptides so as to increase the accuracy scores. Likewise, if I'm taking alcohol dehydrogenase beta 2, coming under the same class, alcohol dehydrogenase 7 and alcohol dehydrogenase beta 2. It is also 375 amino acid, very close to 374 amino acid of alcohol dehydrogenase 7. Here also it gives me 25 peptides, greater than 500 Dalton. See, 26 and 25 also, very close. But when I'm checking the like peptide fragments, the size of the peptide fragment, see, 885, 580, only two peptides are coming exactly close. See, even within the closely related peptide protein sequences, only two peptides are overlapping as a class of their similarity, which belongs to the alcohol dehydrogenase class. Okay, so now you understand like how much significant it is there to even to discriminate between two functionally related like proteins which are coming under the same clade. Okay, evolutionary too much related. The clade. Clade is the meaning of that. Okay? So two closely related proteins yet only two peptides match. That is the beauty of this entire strategy. Okay, mass spectroscopy. So see, like if I discuss the advantages and disadvantages of this methodology, there are few very important advantages. First one is the like determination of molecular weight of the amino acid sequence. Yeah, you will claim that the sir we can get by a protein sequence also. If I just go for sequencing, but there you need to separately go it. Here you can detect the entire part, detect the protein function, get its molecular weight, amino acid sequence, all at the same time. Okay, very precisely. So that's the beauty of this entire methodology. Moreover, if during the cellular like post-translational modifications, if some weight is changed, even by a bit, that also is caught here, because it is very efficiently doing. Okay, so that's the entire advantages of this method. Like further, like for multiple proteins are mixed together, which normally happens. Okay, so in a cell, if multiple proteins turn up together, we'll get the protein fragments, all of them together, and that will make it like we can easily catch or we can easily detect all of these fragments all at the same time. Okay, so that we can discriminate them further. That this current sample was a mixture of multiple different proteins as going with the results. However, there are some disadvantages as well. Like it's the machine or the apparatus is very costly. The equipment is very costly. Okay. Moreover, it requires sequence database which needs to be constantly updated. Okay, whatever the protein fragments coming up, you need to always update the database. Okay, so this is the entire instrument which is doing all this. Okay, the device to do mass spectroscopy, matrix assisted laser disruption ionization, and the TF is the time of flight. Okay, so it is using it. As we discuss further, see, so peptides are charged with positive ions and post-analyzing tube with a magnetic field. And then it's analyzed under the gas phase. So they are moving together towards the detector. Then the detector catches the intensity of these peptide fragments. So accordingly, it calculates the count also, how many counts are present. Okay, the mass to charge ratio, see, M by Z. This is the entire summary that this is used for identifying and characterizing the chemical compounds, especially the proteins. Okay. Tell me the right answer. Each component of a mass spectroscope is responsible for separating ions based on their mass to charge ratio. Very simple detector. What information can be obtained from a mass spectrum? Molecular weight only. See, elemental composition we cannot exactly find out. Okay, structural characteristics we don't know. We're only getting the peptides. The protein structure is already broken down into fragments. All of the above answer is already not correct. Okay, so the answer is the A only. What is the right answer? It is false. Because we are identifying the protein based on mass to charge. Our objective is not to find out the mass to charge ratio only. Okay, that's just a strategy to find out the peptide fragments or discriminate them. Alright. Now we are going to the second phase of this session. Now we are extending it further, see. We are doing exactly like using SDS page, see. Then we are taken it from a research paper, see the trypsin deletion you are doing, okay. You are doing the mass spectroscopy, MS data is sent to the mascot server. You get the data, see, that it belongs to this. Molecular weight search, the mouse which is there, is then done for the all the peptide fragments coming up. See. If it's significant was at least two peptides should match, then it is considered significant. So it's associated with the p-value score, the p probability score. Okay. So besides this, what we do is, see, we also check the function of the desired sequence in Unicode, just to further confirm it, because you are getting some segments. So what is the functional detail of this segment? Can I decode it further? Structural check. If I am able to get the complete sequence or at least close to it, can I build its model or can I decode its structure to then check the structural details of this protein structure to get extend the function? Okay, or the evolutionary check. If I am able to, I show two peptide sequences, alcohol dehydrogenase 2 and alcohol dehydrogenase beta 7. So if I am able to decode it further, can I use evolutionary phylogenetic details? Okay. You see, I did in my research paper only. See, I considered 546 sequence dataset within this length. Enterobacteriaceae. See, I also compared it with the 76 sequence Bacillus subtilis within this dataset, set B. Compare this with physical, chemical, and secondary structure analysis. Then I did MegaX, phylogenetic analysis. See, all these different classes of the species are present here. These are termed as clades, clade 1, clade 2, like that. Each of the classes. Each clade is colored differently here. Moreover, if I am knowing that this segment belongs to that class, can I use it as a motif? Okay, it may be the motive for that class. So can I do that further check? So it is use of searching the required functional motifs in a protein sequence. MEME is the server. MEME, that is the server used there. See? So those of you are interested can search this server again for their own peptide sequences. You can either enter one sequence or multiple sequences together, and then it will show you exactly the results like this. See, here are the sequences I may have entered there. All of them were fitting in the same class. So see, I got three different motifs coming up here. Right. Now see the multi-top, I'm discussing it further, how to do it further. See, we did this complete part. Now the computer search. See, major reason is when the results are coming incorrect. See, peptide mass fingerprinting requires a specific protein only. You understand? We require a specific protein. But what happens? We don't get the same protein only, highly pure protein. We don't get it. Multiple proteins stand up in a mixture. Even though we try to use get the sample free of contaminants, but even then it happens. See, and lastly, what happens? I'll get a mixture of multiple proteins, generating multiple peptide fragments, which finally result into several different bands together. See? Okay, so giving wrong answer. The protein sequence must have a significant homology with other sequences. Okay, so at least it should be finally I can be able to discriminate. If something new turns up, then I need to do further homework, further checks as I discuss further. Okay, it may be difficult to just find out only based on one or two motives or one or two like conserved fragments. It may be difficult. Because the probability may come only 0.5 to zero point 0.6. That may be because of the contamination. Alright. So it becomes doubtful like should I believe the results or not? So I need to do further check. Okay. Only I'm doing computer search as well. I'm repeating the similar part in the computer. So generating the theoretical proteolytic peptides, peptide fragments. And then again doing replicating the same search. Here I am doing with the masses granted by the experiment, mass spectroscopy or the multi-top machine. Here I am doing this entire thing in the system. Okay, getting the computational masses. So proteases here cut at the defined sites. As we discussed, C terminal of lysine or arginine. Okay, so this is termed as peptide mass fingerprint for a protein. So we download the protein sequence database, pick a protease, you can choose any protease. Normally trypsin is used. Okay, determine cleavage sites where it will cleave and generate the peptide fragments yourself if you are doing it in the system. Okay, now we consider one more thing. As because we are considering the masses, it becomes important to analyze the mass of every amino acid. But the mass of amino acid it depends on the pH at which the protein was functional, wherein the protons also turn up. Okay, so I add the hydrogen there. So I should neutralize the charges. Because it will change the mass, overall mass of the protein, leading to substantial change. Okay. So mostly it's the search is the molecular weight search. Developed by metric science. So this entirely part is genome-based peptide fingerprint scanning. So for this, several databases are extensively used and updated. These databases include NCBI, NR, non-redundant database, okay, which is a GenBank, EMBL, DDBJ, several different parts. Besides this, Swiss plot is more preferred, like because it is manually annotated system. Okay. Further, our OWL database, which is comprising see here like Swiss plot, PIR, translated GenBank and PDB database. Besides everything, the EST database, which we have already like discussed a few lectures back, that is Express Sequence Tag. Okay, but only trouble here is that they are highly redundant. These sequences are highly redundant, so they can be placed at several points in the DNA, because of which the quality which is there, like exactly determining the position, exactly discriminating the function of a sequence is very low. Okay, that is the only trouble here. Besides that, okay, at least what is the advantage here? We are able to get some information at least, if possible. So we are trying to use every single database, every single possible option is available to us so as to decode the function. Yeah. This is like database mascot search page. Here you can enter all the molecular weights you are getting resulting from mass spectroscopy. Once you feed it here, feed this data here, you can also enter fixed modifications like some changes have happened. These changes, as we discussed, could have happened during the post-translational part. And all these changes are like simply ascribed to consider the molecular weight change, which can be peculiar and very important to properly discriminate between the few peptides. Okay, so to make the mass search properly and efficient, properly done, we are considering all this. See? And one more thing is no, mostly considering the monoisotropic mass, which is like the mass, atomic mass of the most abundant or the most prevalent isotopes. Okay. But if I consider the average mass, that also can be done, considering all different masses of that chemical components. See, this is the complete list of monoisotopic masses which I'm showing here. See? If you see here, isoleucine and leucine exactly same mass as obvious. Okay. See, to see the results, now we entered some results. So now how the results page comes up. See, if you see here access number, this could be these proteins. See, and the score and the mass here. See, upper results. If you compare it with the, see, just like the blast result come exactly like the rest, but there the e value is there. Here no e value. It's just giving the score and the masses. See, moreover, like see, mass to score is higher. See, 618. Calculated PA score P 5.823. Sequence score 24%. These are the which are the matched peptides here. The matched peptides are shown in red here. See, and this is the cells link which is coming up. See, the input data was there, this is matching to this complete protein sequence. And here the peptides are here. See, these peptides are matching exactly. This is the super part. Besides showing that this is matching with this kind of a conserved peptide segments, it is also telling you that the protein sequence, the function, and along with several other details. Okay. So see the major applications is phosphorylated proteins as we discussed earlier. Post-translational modifications, if they have happened, can be easily caught here. Splice variants, see, if multiple different proteins come out of the same protein sequence because of alternative splicing, the splice variants can be effectively caught here. Because multiple different bands will come in, like proteins with disulfide bridges, again leading to the weight change or any other change. See, they can be properly caught here in peptide mass fingerprint. Okay, so it is termed as peptide mass fingerprint. Okay, it's like the human fingerprints, very conserved. Okay, just like the protein or the peptide mass fingerprinting. Okay, so in this part we have learned several different algorithms and computational tools to analyze the protein sequences and get their functions effectively. Okay, so we remember learned about the like Phylogenetic part or MEME tool. We are seeing the motifs or finding out the motifs that the conserved part conserved across several different sequences. I shown you the data set of 546 sequences. Okay. Besides that, we seen the Mose weight search, how it is done, like monoisotopic masses and all those things. Okay. Now let us discuss the self-assessment questions with respect to this part. Which of the following is true regarding multitop mass spectroscopy? Okay, it is a technique used for DNA sequencing? Of course not. It can also use of laser desorption and ionization. It makes sense. It measures the mass to charge ratio of ions. That is not the ultimate objective here. Okay, so it is incorrect. It is primarily used for protein crystallization. That is of course wrong here. So the correct answer is second part. B. Okay. What is the goal of motif prediction in computational biology? To identify conserved regions within a single DNA sequence. It is not the ultimate goal. Yes, it is partially correct. Conserved regions, but is it a single DNA sequence? Okay, that looks a bit dilemma. to predict the conserved secondary structure of a protein. That is not the right answer. To determine the function of a protein based on its sequence. That is also not the right answer. Because its objective is to find out the motive. To identify recurring patterns or motifs across a set of DNA or protein sequence. Yes, this makes sense. Because if the recurring DNA or protein sequence involves some of the related proteins, the motifs or the conserved patterns will be overlapping each other. So as I shown you in the diagram, we can exactly discriminate the functionally like significant clusters of proteins which are functionally similar. Okay, that makes sense. Now let us discuss the terminal questions here. Many prediction algorithms utilize statistical and computational methods to search and characterize the conserved motifs within the biological sequences. True or false? Of course it is true. Okay, then think the case when a too long motif sequence need to be considered. Now this is a good question. See, if a motif is very long, there's a high chance that several different the segments or the motif segments may match abruptly. Okay, which can lead to overfitting. Yes, overfitting of the data will be taking place. So the complexity, because sequence complexity is not at all there. Okay, because these things will be redundant. So the will many different short motives will be fitting to it. So the accuracy will decrease drastically. Okay. Now see, towards the end we will be discussing like phylogenetic profiling and protein-protein interaction networks in more detail. But before we go further into this concept, we'll be discussing about the protein structures or how to what are the different columns present in the protein structure. This will allow us to properly understand or analyze the data coming up in the later sessions. See the PDB data we'll be discussing here. It includes understand that it includes 15 columns. Okay, these 15 columns are specially meant for showing different atoms. Okay, different what all amino acids are there, how their connection looks like, what all different ligands are there, and what is the coordinate information and all the other details. Okay, these 15 columns do not directly encode the BCC lattice or the BCC matrix is used to solve the crystallographic or the NMR data. Okay, which is through which the structure was solved. All the data is separately encoded over this crystallographic information. This is present at the end. Okay, first six columns, columns 1 to 6 encode the residue type. Okay, if it is atom, simple any atom, and the okay all these parts as atom. Head to atom is specially meant for ligand. Then the header to show the important parts. The compound, what is that compound altogether? For which the atom is denoted. Sequence residue and the connect. Show the important bond. Okay, then atom serial number. 5 columns specially meant for that. It goes from one to the very end, including the ligand atoms also. Then atom name, 13 to 16, O, C alpha, C beta like that. Then alternate location. Sometimes what happens if a atom is there, that atom can be presented two places within the crystal, within the NMR solution. So what happens? This data, which diffraction data or NMR data, found that atom was found at two different positions. Okay, assigned to the probability score. We'll be discussing that here. So we take the one with the higher probability and discard the other one. But we show both the entries in the structure data. Okay. Then the residue name. Okay, what is that amino acid altogether? Chain identifier. Now you understand that protein structure includes multiple chains. It's a multimer, okay, multimeric protein. So A, B, C like that, or 1, 2, 3 also 0. Like any different kind of number can be given here. Okay. Now see the leftover part. Residue sequence number C, 23 to 26 columns. Okay, what is that number of the amino acid here? How many for the number of amino acids? It shows the amino acid number. Earlier it was the atomic number for all the atoms present in the protein. Now code for insertion of residues, XYZ coordinates here. These are simple Cartesian coordinates. Then followed by occupancy. This occupancy column shows the probability. Means if two atoms are present at two distinct positions, two different positions, what is their occupancy? Okay. Temperature factor is also known as B factor, Debye-Waller factor. This just shows the how much is the thermal motion or the like, atomic displacement here. Higher is this score means that atom was found to be more displaced from its original position. Okay, so if we are looking for a conserved part which is not always present at its own atomic place, we need to skip such atoms. Okay, and take the other atom if the two different atom positions are there. Element symbol. Lastly, what is that element all about? What is the row is meaning to? Okay. So see whatever we have discussed regarding the PDB format or its 12 columns. See that's how it looks like. This is like exemplary data here. Atom name. See all the atoms present here. Then 7 to 11. See this is the right line part I was telling. All these atom numbers. What is this atom? What is that atom? Like this atom here. N, C alpha, C O, C beta 1, like that. Understand the peculiar thing here? All the amino acids starts with nitrogen. The first amino acid methionine. In the next column, methionine, glutamic acid. All are starting with N. See? First amino acid position is 1. Second row is 9. Likewise it goes. Okay, this is the chain A. Protein is a multimeric structure, so it can have multiple chains. Each chain can have numerous amino acids. Okay, and this is the residue number. What is that amino acid number? So 1, 2, like that. Each amino acid number starts with nitrogen. See? Subsequent to it is like XYZ3 coordinate. So see 32.632 minus 11.712, 53.84. These three like Cartesian coordinates XYZ indicate the position of nitrogen for methionine chain A. Okay. And what is the probability that they are exactly found, that the atom is exactly found at that place? 1. Occupancy is 1 here. Always it is found at exactly the same place. See here 0.9307. Two positions are there, C beta 1, C beta 2. Now if I ask you which one you will consider, of course the higher one. You will take the C beta 1. See? I'll take this part to be correct and delete the other line. Understand that this is the initial data given by the system. So it is there. But when we do it for the analysis, many kinds of analysis automatically take the higher occupancy part. But if it is not considered, understand that you need to manually delete the second line, C beta 2, because the occupancy is less. Okay. See where the B factor is also high, D by Waller factor, B factor is also high. So formula is 8 pi square by cube R square. See? This radius R is shown here as U also. Okay. Now we'll discuss the phylogenetic profiles to predict the protein function. Normally what happens, we build the sequence data sets of DNA or protein. So here what happens, if we are considering that multiple sequences are aligned together with at least sharing a 30% sequence similarity, then what we believe that they are functionally like a group together. and if somehow they're fitting within the same clade also, either because of the species like linkage, or because of the functional linkage. Okay, so if it comes that way, then what we believe, they are functionally connected. Okay, so now to take this discussion further, if we also believe that they are functionally connected, okay, so if the functional connection keep on coming every time for multiple different proteins together, like same way, what it means, they are always found to be connected. Means they are evolving together. Such proteins are termed as co-evolving proteins. Okay, their evolution has always been in a correlated fashion. Co-evolving protein pairs. So if a mutation comes up in one protein, like the compensatory mutation turns up in the other protein, so that their function does not get disturbed. Okay. See now, to take this discussion further, phylogeny is very efficient tool to detect the gene pairs that are simultaneously present or absent across a number of genomes. Okay, and this is done through evolutionary analysis. So now see like it detects the presence or absence across the different genomes. So they are likely to be interacting proteins because they are somehow connected with each other. Now, what is that exact connection? We can find out by like scrutinizing their pathways, how they are mutually connected. But their connection is certainly there. Okay, so they are mostly interacting protein pairs. Okay, so see here like genes encoding interacting proteins. I'm just calling it as genes. Okay, gene 1, gene 2, gene 3, gene 4 present in genome A, B, C, D, E. I'm finding that genome A, these four proteins are there, see, of which only one protein is present in genome B and genome D also. Alright, understand that. But gene 1 is present in A, C, E genome. Gene 3 is also present in A, C, E. Gene 4 is present in A, B, and D. Now, did you find some discrimination factor here? Wherever gene 1 is present in particular genomes, gene 3 is also present that way. So, what it shows? It shows that their gene 1 and gene 3 are always acting as like a interlinked partner in all the genomes considered here, the 5 genomes. Likewise, the gene 2 and gene 4 here, see, the present in genome A, B, and D here are like found to be interacting protein pairs. Now, what is the exact pathway they are connected? What are the exact details of all that? We need to scrutinize that further. But that information we are easily getting by analyzing this phylogeny. See here, like I am calling four different species here: E. coli, Saccharomyces cerevisiae, okay, B. burgdorferi, and Helicobacter pylori. See, represented as EC, SC, BB, and HP. See, protein 1 to 7. Okay, these proteins are presented, these proteins are presented, these proteins are present here. But when I'm considering all of them, see, this phylogeny, see, it shows wherever protein 2 and 7 are there, they're always connected. See, this phylogenetic profile, what are the phylogeny built for them? I'm connecting for all the 7 proteins, I'm connecting this way, 0 or 1. Presence or absence. 0 indicate absence, 1 indicate presence. I'm connecting building the channels, how they are mutually connected. Okay, overlapping parts. P100, 101. P4 and P1. See? P4 and P1. See? Taking the maximum similarity. Just like the maximum likelihood function works in the case of phylogeny. Exactly the same way I'm working out here. See? P2 and P7 group. Likewise, see? And P3 and P6. See? P1 and P5, see, lateral connection is there, but see, P2 and P7 and P3 or P6, see, are highly connected, see, handling on their function to the same path, some pathway, see, they are connected somehow in some pathway. Okay? See? Now this is a diagram I am taking from some research paper. Here multiple different proteins presence or absence is shown here for several different species. And this is the beauty of phylogenetic profile. Okay. So if you now what is the limitation here? I hope you will be able to understand now. If multiple different proteins turn up, okay, can you figure out where what is present and what is the role or how these multiple proteins are functioning together? We cannot find out because multiple proteins are present together. Now they may be present in several different pathways. Okay, if five proteins are present common, are these five proteins are interlinked within the same pathway or multiple different pathways which may be connected, which may not be connected. Okay, so to understand this part properly, what is the major requirement? We want that all these proteins, whatever we are taking, should be less in number, very small number of protein. Okay, which you are doubting. Moreover, the species is there that we can take as much as we want, but the protein number should be in less number of proteins, so that we can find out some correlation. Now, an important question turns up here. If you are taking a less number of proteins, what should be the protein size should consider? I hope you understand. What should be the proteins I should consider for the analysis? Okay. So for that, understand that if you are considering building up phylogeny, consider the proteins which are like found to be similar within a clade or within close clades, and which are like you are doubting their function. Are they present in the same function or not? You have already checked different databases, different things, but there like confirmation is still you are skeptical about that. Like should you trust the results or not? So you should take those protein pairs and then check it further, especially when they are affecting the mutations. Okay. So this discussion brings us together to another concept, which is termed as gene order. That is order of arrangement of genes on a chromosome, which is normally poorly conserved. Okay, but still, if you find that some genes are particularly like linked in an operon, those operons normally exist in prokaryotic organisms. Like the genes show conserved across divergent genomes. Okay, so that order is conserved. So what is an indicator of? It is the indicator of that these genes are physically coupled. They may participate in a protein complex which is forming at the same time to work together on certain important biochemical function. See here, like in this slide, I'm mentioning the protein-protein interaction (PPI) and the biological pathways databases. You can properly analyze the phylogenetic correlation. Okay, so to decode the like a set of coupled genes again. Okay, BioGrid, this Mint database. The KEG is the Kyoto Encyclopedia of Genes and Genomes, the Japanese server. See, the EcoPsych. This is basically meant for E. coli. So the several different research teams have already been working on E. coli. And this is a string database, which I personally love. See here, what is this major advantage in string database is that it is much more like sophisticated database, much more evolved database over all of them. Okay, whatever the details you find here are normally not at all available in those databases using statistical scores, like the way you are able to correlate or the way it is able to integrate with several different other tools. Okay, here if you are putting some genes or proteins, you can always build their network and represent it the way you want. Okay, see what is the part you are supposed to do here? You are supposed to just upload the gene names or the protein names in all of them, and in the result, it will be giving you proper network. It's just like graph, like five proteins A, B, C, D, the connection, mutual connections. Okay, so just like the graph theory part, concepts work here also. Graph theory, like all the proteins will be termed as nodes, genes will be termed as nodes, and the connection between them will be termed as an interaction. Under what condition, what things, constraints the interaction goes on, that is different. Okay, but that interaction is there. Okay. See, like if I just extend the results to a large number of proteins, and I ask you this question: if I'm taking so many proteins and I'm building the like so too many different set of proteins which are much more connected as a set here. See, one set which is highly connected here, other set highly connected here in this graph, one set connected here, one more set here, and one more set here. There are five sets altogether. Amongst these five sets, I am finding so many different connections which are found to be like experimentally validated or just resulted based on the homology. Now understand that how many of them make biological significance. Maybe some of them. Okay, but how to find out? This brings up to a very important part. If I am predicting the network analysis and functional annotation through phylogeny, through all of these, putting them together in a network, protein-protein interaction analysis, PPI network analysis. Okay, I'm finding doing all that. I'm able to assign the scores. Okay, like how many times I'm finding the genes to be always connected. I'm getting everything. Okay, so but can I do that that interaction to be confirmed with additional databases? Okay, as I listed earlier, additional interaction data sets need to be considered. That is also done. Okay, but how should I validate it further? Or all these predictions are like just confirmed with some scores. But why should I believe that? I'm not skeptical, but still you should be somewhat skeptical to do all this because this is so much details, so much data just assigned with some scores, which may be some garbage numbers or arbitrary numbers. Why should we believe it? Okay, so to further do it, we validate or to verify the results using some wet lab experiment. Okay, if we are finding that these two proteins are always connected together, okay, just like the ribosome proteins have several different protein like subunits. If I am mutating some of them, is the ribosome functioning, overall ribosome functioning? If not, then I believe that yes, even if the other subunit was present, the failure of this subunit fails the entire system. The entire system fails. Means there's a properly governed like a protein-protein interaction network going on in this system. Okay, you can trust that. Okay, so whatever the prediction you did earlier was right. Okay, you can come to a conclusion or you can validate your results. Okay, once you are able to do all these the predictions, verification using the experiments, and then you are free to publish the results. Okay, because you have done a very good experiment. Okay, now come to the self-assessment question. Which of the following best describes the purpose of phylogenetic profiles in a protein analysis? Determining 3D structure of proteins? Of course not. Analyzing protein-protein interaction networks. Is it giving you interaction networks? Okay, that is not at all giving. Predicting protein functions based on evolutionary conservation. It makes sense. Identifying post-translational modifications in proteins. That is of course not. Okay, so option C is the right answer here. Which information, what information can be inferred from a phylogenetic profile analysis? Protein-protein interaction partners. Now see the first question is twisted here again. Post-translational modifications, protein 3D structure. relationships and the similarity between proteins. This part is again the right answer here. D. Option D is the right answer here. Okay. The question is just tweaked. Okay. But I hope like you are very confident to get the required answer. In the first question, C is the right answer. In the second question, D is the right answer. Okay. Now come here. Last question, see. Inferring the evolutionary relationship between proteins is the primary application of phylogenetic analysis. That's what we discussed in the last question. So I'm sure that you are getting the right answer true. How it is helping to find the reliable interacting protein partners for a candidate proteins. Can it find which is the reliable interacting protein partners? I hope you understand that I'm somehow like inferring with the protein-protein interaction networks, the protein partners. Okay, when we are building phylogeny, we are saying that they're like a functionally, functionally sharing like a close similarity. But are they like, interlinked together? We are not at all saying that way. But can it be like how is it helpful? Helping us to find the reliable interacting protein partners. So answer is yes, it is indeed helping us. So if you are having a large number of sequences, you can use phylogenetic to club them to a like much more like a highly related number of sequences. Okay, which are evolutionarily linked. So then you can build two phylogenetic. Once you are done, okay, then you can analyze it further using protein-protein interaction networks. Okay, so phylogeny builds up a basis to analyze the protein interaction network data sets. Okay, so it's very useful. Okay. Thank you so much for your time and patience.